One of my collaborators literally treated people with depression with the Mediterranean diet. It's called the SMILES trial. You can take the microbiome from somebody with depression, transfer it into an animal model of depression, and transfer depressive-like behavior. So it's like screaming at me, where like maybe microbes are the reason in some people that they have depressive-like behavior. There's all these signals, even if 10% of them are true. We could be curing depression in certain people. We could be preventing Parkinson's.
All right. Hey there, everybody. On this episode of On the Rise, we're lucky to have Phillip Strandwitz. He's the CEO and founder of Holobiome, a startup that's solving the complexity of the human gut microbiome and making drugs and consumer products out of that. With a PhD in biology from Northeastern University, Phillip is a founder with deep expertise in the field. He is committed to translating the science of the human microbiome into next-gen probiotics and targeted solutions for whole body health. Strandwitz specializes in microbiomes, and more specifically, the approximately 10 trillion bacteria that live in the human gut. He's been quoted saying it works out to be about 4 to 5 pounds of bacteria in every human, and he just fell in love with the topic. Now at Holobiome, it's a B2B platform in biotech that's unraveling the complexity of the human gut microbiome to discover and develop products to fix health. The first industry they're going to transform is the $80 billion consumer probiotic industry, which they claim to be largely consisting of snake oil. Next up is pharma. I'm so excited to have you on the podcast today and to dive into the fascinating world of microbiome with you, Phillip, but also to hear a bit about your startup and on the rise story. So super pleased to have you here. Welcome to the podcast.
Awesome. Yeah, it's great to be here as well. I think we're going to have a lot of fun.
Great. Yeah, so for a little bit of context, it's been a few months since we met at an event hosted by Alan. I included a lot of founders in a venture capital portfolio, but your work really struck me as unique, and the positive impact and outcomes of your work can be so clear. I definitely want to hear about all the research that you and your team are doing on the human microbiome space. But maybe first off, you could give us a little brief background about yourself and what even piqued your interest in biology in the first place.
Yeah, for sure. So I originally come from the Midwest. So come from the land of cows in Cornfield, Wisconsin. So grew up in a small town, ended up going to a relatively small state school, but have always been interested in biology ever since I was a little kid. And my mom really focused on landscaping. And when I was younger, what I loved to do was to like go out to the yard, see a rock, turn it over, and look to see what was under the rock. And, on this like perpetual quest for salamanders and snakes and whatever things, that were there, but I couldn't quite see them. And so it started with that inherent curiosity. As I went to college, I became very interested into the field of microbiology. So studying the things that we actually can't see with our eyes. And so we need microscopes and various things to take a look at it. And when I was at the University of Wisconsin-La Crosse, really became just fascinated with this idea that we have this ecosystem of microbes that live around us. We're starting to understand that they exist. We largely don't know what they're doing. And there was this concept of uncultivability, where you could take, say, a sample from like soil, you could put it under a microscope, you'd see 100 cells. But if you tried to actually grow those cells in the laboratory, only like 1% of them could grow. This is known as the Great Plate Count anomaly. And the idea here for me was like, wow, this is pretty cool. We know we live in a microbial world, but we have never accessed the microbes. It'd be pretty sweet if I could continue to research this. So I tried to find the top labs in the world that were trying to crack this question of, all right, microbiology, you're a field. As a lot of fields in science go, you have your model organism, you study that, but that model organism isn't representative of actually the world. So long story short, I left the land, the cows and cornfield, to come out to Boston, Massachusetts. Fortunately, it was during the time when, you had smartphones and you had Google Maps, otherwise I would have been totally lost. And I joined Kim Lewis's lab at Northeastern University. And the idea here was he's been studying this problem, the Great Plate Count anomaly, for many years. And his interest was to try and isolate microbes from soil to then uncover antibiotics. So a lot of like the drugs that people take today, they're actually sourced from bacteria. And again, if you have all of those sourced from that 1%, get the 99, there's probably a lot of good that you could do, right? So I joined his lab in 2011, was convinced I was going to be an antibiotic hunter, and to join the best antibiotic lab in America to do that was, really on a cool track. And then a pivot happened in which Kim's group got one of the first grants from the Human Microbiome Project. So the NIH, National Institute of Health, it funds a lot of research. Early days of microbiome, which we'll talk about in just a minute, it really just started with an infusion of capital by the NIH, where we started to understand that microbes were on and in us. We didn't know anything about them. And he had one of the first research grants to try and isolate bacteria from people. So not from soil, but from people, to try and get these microbes in the laboratory, understand what they're doing, and potentially connect the dots with health. So that was like my first pivot, I'd say. And I've had many pivots and many different things that I've tried, but I stopped being an antibiotic hunter. I said, hey, when are you there at the genesis of a field? I became instead a poop scientist, and that brought me on a very different path. And to cut to the chase there, during the PhD at Northeastern, as the field of microbiome continued to grow, it became very, very apparent to me that we were going to find cures if we could understand our microbes better. And rather than staying in academia and largely publishing, I was very, very motivated to try and take the knowledge that I generated and some of my colleagues generated and turn it into an ecosystem where we could create products. And where it's not just a PDF you publish in Nature and that's that. You instead actually do the research to have, products where you can test hypotheses, you can do good, you can empower people with biology. And that's what led me to Holobiome. So I'll take a pause there, but happy to answer any questions about that.
Well, that's fantastic. I got to first start by saying I'm also from the land of corns and cows, but your neighboring state in Minnesota. So I totally understand that. And actually, it's funny, I actually got interested in biology from working in a plant biology lab way back in high school, but didn't obviously stay as dedicated to that as you did. And then you made such a big pivot from there to Boston into microbiomes and this company piece. What did the community look like to you when you were making that decision? How big of a community was there? You said it was growing. Tell me about that.
Yeah, so I would actually say Wisconsin, and when you think biotech, there's mainly two hubs. So there's Boston, there's San Francisco, and San Diego is doing quite a bit better as well in that too, and now New York and Raleigh, North Carolina. But there just isn't a lot of biotech in the Midwest. And so while that's where I got my spark of curiosity, it was really, clear to me that if I wanted to try and do deeper research, maybe get in the arena of starting a biotech company, I had to leave. And to give you an example, where I was going to school in La Crosse, there was really one internship in microbiology as a whole, and it was like a water testing laboratory. So I still remember my days of going into this water testing laboratory, and I would get like cotton swabs from pigs, and I would look to see if I could isolate bad bacteria from them. We would also get like Brussels sprouts and broccoli sprouts and look for like fecal bacteria in them from like, produce producers. And then we also got like water when cities would flood. So I was in this like very small lab with a tiny window, like doing like that type of work. And you compare that to like coming out to Boston with all the universities and internships and hospitals and everything, like it's night and day in terms of, just exploring curiosities. So while, the foundations were good for curiosity in Wisconsin, I had to come out of Boston to expand the community. And I think that's just, a lesson for anybody thinking of getting into a field. Like it matters to be there where magic's happening. And like that's what obviously made Silicon Valley so magical. You walk down the street of Kendall Square, which is like the Biotech Hub of Boston and Cambridge, like you just see people, it's crazy. And there's like restaurants you can't even go to have a private meeting because there's so many VCs that are there that like they pay attention to like who you're talking to. So yeah, community is really important for sure. And, I think I found mine in Boston, and I can, talk more about some of the groups that have helped me as well.
Yeah, Philip, Steve Jobs has this quote that goes along the lines of, it's only in hindsight where we can connect the dots. And when I was hearing you talk about growing up in Wisconsin and how that led to where you are today, but I'm curious, when you're in the middle of the journey, how did you feel and how did you navigate the uncertainty and how did you follow your curiosity?
Yeah, it's a really good question. And so the way I've thought about things, and I've talked to a lot of people about this as well, is, there's a few bins of like life. So bin one is, are you happy? Like whatever you're doing. Like if you're happy, you basically won the game. And just do more of that because that's probably the key feeling to chase. The second is, are you healthy? Of course, and that's a harder one to chase. The other, which is tied in part to happiness for some, but not everybody, is do you have satisfaction in what you do and you feel like you have purpose? And so when I was going through undergrad and even parts of grad school, I was definitely happy. I had great friends, great families. My wife is fantastic and I met her in Boston. And so that happiness, I've sort of had throughout life, which I'm very fortunate to have. From a health perspective, I've fortunately been okay there too. But then from like a purpose and satisfaction perspective, this really gets me because I've never felt satisfied with what I do professionally, even today. And, that to me was the driver of I got to mix things up. So in Wisconsin, as an example, I had a little landscaping company throughout my undergrad and I loved it. Like I was outside, it was sunny, I was like tan, not like this pale scientist you see today. And I was employing friends, like it was, great. But it just wasn't scratching that satisfaction itch. And then when I found science and microbiology, I got my first like taste of it. I'm like, you know what? This would be really neat. If you could have something that's really intellectually challenging, that could have a lot of meaning, you could challenge dogmas. So microbiology, to me, I saw a problem. Again, this concept of we know there's a lot of microbes, we haven't really studied them well. So I chased that, thinking that it would find me satisfaction. I then found the microbiome, which, this was an opportunity because it was a new field. To me, that's really cool if you can be there at the beginning because you're going to learn a ton. And learning is tied to my satisfaction too. But then what I've come to recognize is, and what I hope will satisfy me, where we could be part of a team or facilitate the research to find a cure for something.
Yeah.
And I don't care if a cure is a drug. I don't care if a cure is a new food. If you cure something to me, that's about as satisfying as a challenge could possibly be from science. And there's an example that I use regularly because it inspired me tremendously, was Genzyme, one of the greatest biotech companies. There was a disease called Gaucher, where basically kids had a genetic disorder, they weren't producing a certain enzyme, and it resulted in them dying. And researchers identified at, I think it was Harvard actually, but Genzyme was a group to develop it, where they found the source of the enzyme. And at the time, this was in early days, before synbio really was a thing, the source of the enzyme was human placenta. And what they ended up doing, long story short, is they somehow built a cold chain without the internet to harvest most of the placentas in America, to then bring them to processing plants to purify this protein, to then give to these kids who required something like 10,000 placentas for one treatment. This is mind-boggling, right? But where I'm getting you to is like, you have purpose, right? So in this case, you're the Genzyme team chasing something that they knew would have meaning. They knew it would work. It was really just like a process of getting there. And the stories that are told of Genzyme today of people who work there is they actually invited all those patients back. So when they created that drug, they would regularly bring the families that literally wouldn't exist unless they did that, unless they moved mountains and they challenged dogmas. And so I look at that as inspiration of something that I could chase. I hope if we get there, when we get there, that'll scratch that satisfaction itch. But anyway, so that's sort of been my driving force. And I found a lot of people in biotech in particular, so many great people in this field, that there's a similar like incredible frustration for not being satisfied. But I think if you can capture that energy right, That's when you hop on a podcast at 7 p.m. after working 12 hours, right? Because you're just energized to like talk about this stuff.
Ah, that's amazing. I can tell you're so deep in this particular topic, and I think it makes a big difference when people care about what they're doing, and rather than just have things be fast fashion or fast fixes. Along that line, I'm really curious to get your take on sort of the growing interest in prebiotics, probiotics, postbiotics, and how it's being marketed in the field today, and how you think, these can actually make a substantial difference. Like, are these green powders, these prebiotic pills, are they really making differences? What is it going to take to truly solve and cure like life diseases or life problems, ailments?
Yeah, really good question. So let me give a little background on the microbiome first and like where things sit. So when we use this word, the microbiome, we're talking about the microbes that live on and in every human. So you have a couple of different types of microbes. You have bacteria, which is what most people talk about. You have viruses that have evolved to actually target the bacteria, called bacteriophage. You have fungi. You also have archaea, protists, a few different things, depending on the person. And so you have this complex ecosystem that makes up the microbes that you've never been without. And so one way to sort of think of this is, as a field, we didn't know how complex this ecosystem was until sequencing came about. So we were basically in the dark. Around 2011, for the first time, we really started to be able to like swab something and get an idea of what the microbe is. So that to me is pretty fascinating. So, we're only talking about, 12, 13 years to where we've started to get to that point as a field where we understand what's there. So we are a complex superorganism. These microbes are effectively a reflection of you, your experiences, the experiences of humans, the experiences of your loved ones, generational transfer of these microbes. You can look at coprolites, so like fossilized poop, and you can see microbes that look very similar to the microbes living in people today. And these are from thousands of years ago. So humans have always had microbes. And so again, we just started to appreciate how diverse this is. It's sort of like pretend you go to your backyard and there's a veil and you never knew what was behind the veil, but you open it in 2011, and we're still learning some of these things today. And you see this beautiful garden that has hundreds of unique plants. So every person, and we'll focus on the gut, has somewhere between 150 to maybe 250 unique species of bacteria. I want you to think about that like a unique plant in your secret garden that I just brought up, right? So this thing is a reflection of all of that evolution, of all these different lifestyles, the foods you eat, the people you interact with to build this thing. And what we're starting to appreciate is it's really important for the entire human to function. So how to train the immune system, how to stimulate the nervous system in different ways, how to help us digest food, how to protect us from foreign invaders like bad pathogens that ruin spinach, you name it. So this is your garden. All of you have it. Where we sit today, though, is we as a field have started to appreciate that this garden is tied to a lot of stuff. And even if 10% of it is true, this will be one of the greatest discoveries of human health. There will be things that are not real, but if there's 10% of what we're thinking today that's real, this will lead to many cures. But so you have your magical garden. We're starting to appreciate it's valuable, but we're also starting to appreciate that sometimes the gardens are destroyed. So this can be from antibiotic use. So antibiotics, imagine you kill half your garden. This can be from bad food. Imagine you poison, some of those plants. This could be from certain foreign invaders that take over the garden, your weeds or whatever. And so when you have that type of a situation, that's now being tied to human health. So the destruction of this ecosystem is bad. What do you want to do if your garden's destroyed? You want to fix it, right? And what we sit today is you have a whole system of probiotics that's been around for a long time. The diversity of what you can buy is very, very little. Think of it like one or two plants of that entire garden. So yes, it may be a part of the ecosystem, but it actually doesn't have the capability of the entire ecosystem. but marketing teams love to say it does, which drives scientists like us crazy when they do it. And what you really want is you want a ability to fix it, but we just don't have the tools. And so an analogy again from like a garden perspective, like if you're in the States and you've ever like driven to a gas station, you know, sometimes they sell these like shitty plants outside. Pretend that's the only plant you can buy. That's where we live today. You don't have garden settings. So you can't even get the darn plant and put it into the garden because we can't buy it as a society, even though we know it exists. So it's this huge opportunity, right? It's like rediscovering agriculture.
Yeah.
But one part of it is obviously getting the plant back into the garden. So this would be like probiotics. So where you have the live organisms, you put it back into the garden. The next piece is you need to make sure it's fed well, right? You know, all plants, they have different sunlight requirements, different nutrient requirements, you name it. So we also have to understand how food impacts this garden and the specific members within. The problem, again, today is we don't have the ability to put plants into the garden, and we don't understand how food really impacts our microbes as a field. And that's something that's solvable with technology. And so as Holobiome's thinking about this, it's like, yes, we respect there's been some work done in this space, but you have to acknowledge it's radically changed over the last 10 years. And if we really want to capture the power of the microbiome, you got to develop the right tools. And to develop the right tools, you need the right technology and the right team to do it, who is willing to call out the bullshit and say, you know, our North Star is like, we want it to work. So anyways, that's a pathway on, but a little bit of background of like how to think about your microbiome.
Yeah.
I will say various tips that a lot of people ask, like, yes, it's fermented food generally is good, is what we're seeing. Yes, eat vegetables. So the complex fibers within there, yes, the microbes within your gut like it. But, you know, imagine you have like a corn fertilizer and you're trying to grow your corn a cob in your magic garden, as I brought up before. The corn's not there, the fertilizer doesn't do anything. So, you know, there's a couple of just huge gaps that we need to fill. Tech is here. Scientists are motivated to do it. And there's, you know, certainly a path to make that happen. But I'll take a pause there. So that, I hope, catches you up a little bit on like the field of probiotics. It's one plant of a very large garden. Mm-hmm. And I brought up each person as 150 to 250. When you zoom out and look at the entire world, it's closer to 10,000. So we're really like scratching the surface. And then from a food perspective, we just don't know how the food impacts our microbes largely. So anyways, there you go.
Yeah, to extend the metaphor, it sounds like what I'm hearing is there's almost two layers or two hierarchies of the problem that you're trying to tackle. One is you don't even know what's in the garden. We just sort of realize the garden exists and we're trying to find all the other plants and how to get the seeds, how to plant them in everyone. But then there's also the sort of end problem where you touched on how what drives you is finding the cure. Is that sort of how you frame it as well, where part of the problem is the knowledge and the discovery piece, and then the other layer of it is how do you actually take that knowledge and apply it to the real world, like almost translating knowledge to wisdom?
Completely. Yep. So, and I've used a lot of those same words. So like we'll talk about the stuff we're building, but like it's all to get knowledge. And once you have knowledge, then you can develop products. You can develop smart drugs. You can develop smart foods. You can develop next-gen probiotics, but you need the knowledge to do it with the appropriate insight. So spot on. And, you know, we're still learning a lot of this stuff. And I could talk in great detail of like specific components of, you know, these problems. But again, it's no longer a technology problem. It's actually an execution problem and really just a capital problem. And, you know, the motivated teams are there. The path is pretty clear. And it's, you know, an interesting business case now where you have like such obvious biology that needs to be solved. Such obvious biology. And you need to connect that biology with the product. And you need to do that in an ecosystem that isn't only looking at you. So like in the case of biotech, you know, biotech is having a lot of issues right now. And a lot of markets are having a lot of issues. There's also a lot of shiny objects out there right now too, for investors or for society as a whole. So how do you compete with this? And how do you also build an organization that can do both research but also product development in a sustainable way, where you're not hedging all your bets on one thing, which is also the traditional model of biotech. And so our goal is to create that institution that can do the research but can develop the products.
This is
And
awesome.
we've been looking to fund that in a few different ways, including grants, partnerships, and now we're exploring venture.
That's awesome. Phil, I think we have a lot of guests on the podcast who we'd like to call multidimensional. These are people who, it's kind of the opposite of well-rounded, where you're just kind of good at a lot of things. Multidimensional means you're going deep in a couple of really big areas, and you're betting a lot of things on it. You're experts in those areas. I see that in you in a lot of ways, from the stories that you're telling and the way that you're trying to set up Holobiome. It seems like you started off deeply scientific, and now you're talking about the technology being there, sequencing is around, and it's about the execution. It's about setting up the institution. I think it's pretty rare, at least from what I'm seeing, to have someone go straight from school to school into starting your own company. It seems like you've never been a part of an organization. So tell me about like how you made that decision to make a leap and to start something, go off of this and really commit to building this and doing this for multiple years at this time.
Yeah, the first thing I'll say is not just me. So I've had a lot of support over the years of like mentors and people that have guided me. At Northeastern, as an example, I had a very supportive advisor, Kim Lewis, who's one of the co-founders of Holobiome. So he's my PhD advisor, my boss. And he really encourages his students to just think without boundaries. And so there was never a point in my entire interactions with him where he said I couldn't start a company. But there were many other people who did say that. And so, having support like that from a mentor is incredibly important. And, he was, I would say, my first like true mentor.
Mm-hmm.
And I've accumulated many others over the years as well. And I've seen just the people that think in this way can really open doors and facilitate creativity. But, now I say, all right, I have somebody that believes in me. They'll give me a chance. Let's talk about the real world of like going out there. And so, basically launched Holobiome in 2018 was when we did. And prior to that, I had billionaires invite me to their house, shake my hand, tell me they're going to give me $10 million. It fell through. I had numerous grant applications that we submitted get rejected and like horrible comments. I even had a grant reviewer from the NIH tell me Phillip Strandwitz can't possibly start a biotech company. And I'm like, man, you're just a jerk. Like who does that? Like that's just so nasty. But so there was a lot of like difficulty in doing it. But it was again this just like stubbornness of like, I really think this is what's going to keep me satisfied. And my motto too was as long as I was learning as I did it, you really start to just accumulate things. So like microbiome, I spent years studying. I study it constantly still today. And there's like a learning curve that you sort of hit, right? And at some point it starts to level out. And the best way I think to continue up that learning curve is do something different and ideally something complementary. So in this case, I tried to learn biotech and what that ecosystem was. But I once made a slide for an entrepreneurship department. I forgot what it was. It was something like, 200 meetings, 11 failed grant applications, like boom, 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 all sorts of failures. But I learned a lot and I met some incredible people. And to me, like as long as the journey you're on is accumulating, value in that way too, like you'll always be okay. And I also made so many great friends and have met so many people that think differently. And it's just a real treat to even be able to be in the same room with some of these folks. I brought up Genzyme. So Genzyme was an inspiration for me in high school. I am part of the Tremere Foundation, which was started by the CEO of Genzyme, Henry Tremere. And it was a mentorship program. I go to Henry Tremere's house. And he unfortunately passed away. He's a brilliant, pillar of biotech, but it's just like, it's astonishing, just how fortunate I've been. So I am not alone is the takeaway, and never try to do it alone is my lesson. And also just avoid assholes because there's so many of them. And I literally have a list of people that like are assholes and they're just like shunned. And then we have like CEO networks and we all talk. So we share asshole lists. So like, just don't be an asshole. Like it's that simple. Don't be good, be motivated, chase the right thing, and like things will probably work out.
Yeah. I have a doubters list, which is similar. It's like people that doubt me, and then I just keep a record of it. I don't really plan to use it or distribute it, but it's just for record keeping. You touched on the importance of learning, and I have this work in progress theory. I wouldn't say it's 100% truth, but for me, in regards to sort of the explore versus exploit problem, I have this working theory that there's actually no limit to the breadth that you can explore. And so I think of it like if you're driving a car, you have to stick to the streets that exist. And so your turns are limited to the roads that exist. But if you're sort of self-directed, you're kind of on the salt flats or a plain of land where you can go anywhere you want. I'm curious if you like what you think of that, and also how that ties in with your research, along the lines of like, did you have any breakthrough moments, and how you define those and how you think those were generated?
Yeah, it's good. Good commentary. A couple of quick thoughts. One would be, I totally agree. And a question is like, as you wander, like how do you accumulate knowledge in an efficient way? And I think one of the best ways to do that is actually just befriend people that are experts in a respective field. Because like if you do self-discovery of a field or a problem, there's so much noise. And like I guarantee if you ask like people to say, I'm a microbiome expert, 100 people will say they are. But if you challenge them, technically they are not. And so, this can lead you down some really crappy pathways, pun intended, if you're a microbiome company. But so I think, as you're on that quest for exploration, like how do you get there to learn it? Like, is it self-discovery and Googling and like reading? Is it interacting with patients to understand what that problem looks like? Is it interacting with subject matter experts, attending conferences? I think there's ways to do that wandering really efficiently. And then be honest too, like what draws you. Remember those three pillars I brought up before of happiness, health, and satisfaction. So like, what are those things that you uncover are you being pulled towards? And the final statement I'll make too, and we tried to do this, and I just did it with some brilliant people, actually on Tuesday. Brainstorm, but just like wash away the bureaucracy and be like, all right, yeah, if there wasn't a tech transfer office or money wasn't a problem, like what would we actually do? And like one moment for us, which we still like some people really challenge, which is fine. We're a microbiome company. We're building a platform. The general goal of what we seek to do is map how human gut bacteria influence our biology, generate knowledge, as you talked about before. And with that knowledge, we then want to make impact. The dogma for biotech is make impact as a drug, which is going to cost hundreds of millions of dollars to even test one hypothesis. And it's going to take many years to get it there. I'm a believer that you can make impact for health, not as a drug. So what if we improve food? And there's certainly a lot of evidence that food can influence health very, very broadly. Microbes in food are very tightly linked. So Holobiome isn't going through the typical path of we'll only be a drug company that has a single asset that we throw hundreds of millions of dollars on. We bet it all in clinical trials and see if it works. Instead, we're a research entity that can uncover hypotheses and choose a path that we think is best for humans as a whole. And that includes consumer applications as food. It also includes drugs. So, that if I went to my mentors in early days and asked what to do, they all told me to just go drug only. But now you look at the field of personalized health, nutrition, like diet's right there. Supplements, there's certain ones with, pretty good evidence that they can improve health. There's certain diseases that are really hard to run trials for. So how do you ever get that data? Unless you have interventional trials with, some other means, like longevity. Oh, my goodness. How are you ever going to have a longevity drug? Unless you have some like real world monitoring of the interventions that you can then tie to it. So anyways, that's just an example of like, once there are barriers that you'll encounter in the field that you choose to focus on, I think it's really refreshing to think beyond those and find people that can do that too, if that's what draws you.
Yeah. I want to take that analogy of learning and the way you do it personally and at your company one step further. I worked at Apple for a little while, worked on a really interesting team with PhDs, researchers, and they were developing really amazing ways that we could take voice data and productize that. And you're sitting at a really interesting intersection where I saw most of your team PhDs, and you're like you function as researchers at the core, but you're also building products and you're doing research. So I'm curious, like, how do you go about thinking about research and tying that to a product that you can then sell out to consumers, not as a drug, but something that you can get out and test? And so I'm curious how you've structured learning and productizing things at Holobiome, given such a unique intersection that you're in.
Yeah, so how about I give a little bit more detail on what we do? So, I brought up this idea of these magical gardens that we all have, didn't know about, and we got. And we want to fix them, make them better. So to practically do this, you need to have access to the plants first, right? And really no group has access to all of our microbes yet. Nowhere even close. And, this is coming from someone who spent my entire PhD on growing bacteria. So there's three core pillars to what Holobiome does from a research perspective.
Wait, Phillip, sorry, could you clarify real quick? Why is it not? Why do we not have anyone who has all the, what's the right term, microbiota, or the 150 to 250? If it's there in everyone, why can't we get it somehow?
Yeah, this is an example of the technology is there, nobody's executed on it, and we are. And so practically, like bacteria in your gut, they don't like oxygen. So if you would come to my lab, we have these big space bubbles that on the inside, there's no oxygen. So let's say you take a sample from a person and you want to grow their gut bacteria. A lot of microbiologists, they would traditionally grow microbes like at aerobic environments with oxygen, but a lot of bacteria in your gut don't like that. Well, they'll just die. So to isolate microbes, you need to have anaerobic environments and certain specialized equipment to basically facilitate that at scale. So there's a bit of a hardware problem. The next question, and it's very similar to this idea of like, how do you feed your plants? All microbes or mini microbes, they have different nutritional requirements. So sort of like cooking, if you pretend you have 200 plants, you may need a variety of recipes to grow them all. And so there aren't a lot of cookers, if you will, from the microbiome perspective. I was in a lab that was doing a lot of cooking. So hard to grow bacteria in the lab generally. Need to have the recipes, which we don't have a recipe book yet. But then this brings us to the third problem. We may know the microbes exist in humanity, but to get the microbe, you need a sample that has the microbe, right? And so there's not an atlas, a biobank of human samples that we know all the microbes that live within them. So that paints a little bit of a picture. There's a technical issue with it. It's hard. There's cold chain considerations as you get samples from people, and microbes don't like that sometimes. But with that being said, there's really three pillars that we built at Holobiome to solve that problem and many more. So the first problem, again, is if you want to get all the microbes, you need to have samples from people that within those samples, all the microbes live. So what we've built is a donor network. We have around 9,000 people that poop for us for money for a $50 Amazon gift card. We continue to expand upon this. We just completed a very large baby poop collection, actually. And so we were getting like diapers and stuff. It was an experience. But so we have a donor network. So we have people that, just like Genzyme. Remember I told you that story of placentas? We created a cold chain and kits to get samples from people's homes to get to our laboratory so we can get the microbes. So first step is building a donor network of people where we can get bacteria. Of note, in this community, we're expanding to include specific like patient groups and other super healthy individuals that we could source, potentially really interesting microbes and get data. So we have the donor network, and we're getting a lot of data from it, but also the samples. The next part, the second pillar, is we're building a comprehensive collection of all known human gut bacteria. So again, we know there's roughly 10,000 or so, depending on how you define it. We're basically looking within those samples, seeing what lives there, and getting all the unique stuff. And we're creating this into like a refined panel. So it's sort of like, wandering through the jungles and picking the plants you want and bringing them home and cultivating them. So we're building that comprehensive collection of all gut bugs. There's maybe 10 companies in the world that are building culture collections. So we're already
question
in a
here.
very...
Just for myself and probably for a lot of the audience, is this like when you go into a rainforest and you discover a whole new species, or are we discovering known microbiomes that we just haven't seen in humans before?
Oh, this is good, because the answer is we discover new stuff all the time. So again, we get the samples from people. You use sequencing to see what's in there. Sequencing inherently is a technology. There's a depth to it. So how many nucleotides do you sequence basically to get an idea of what lives within there?
Mm-hmm.
So depending on the depth of sequencing you do, pretend like a sample is like a wedding cake. A lot of people do shallow sequencing. So you basically only look at the frosting at the top. You don't even know if it's a chocolate cake. You don't know the fruit maybe on the bottom layer or whatever. So sequencing does give you a look, but it doesn't give you a comprehensive look. And there's things that can be below the limit of detection. So that being said, we get a sample. When we isolate microbes from a person's sample, we almost always find novel species that are not known to science. That to me is so cool, right?
Yeah.
Again, as a kid that likes to flip over rocks to find like salamanders, now like we can find new microbes. And these microbes could be cures for things. They could really help us understand biology and disease. So does that answer your question?
Yeah, perfectly. You've clearly refined your analogies over the course of this company.
Yeah, well, I mean, honestly, it's really complicated. Like, I don't think there's a more complicated field of biology than the microbiome. And it changes with food we eat every day, like in terms of, levels. Everybody's a little different, albeit there's some overlapping functions. All the classical problems of other fields are also here. All right, how does microbes influence neuroscience? Well, what are models to study neuroscience? So, there's all of these things that are just complication upon complication upon complication. So it requires a whole book of analogies, basically.
Wow.
With respect to the donor network, I've heard one criticism of therapeutics and healthcare overall is certain biases towards demographics because the research and the development is disproportional to the true population, whether it's ethnicity, other sort of factors. And I think it's in one anecdote that I've read recently is the Hazda people. They're like this indigenous tribe, and they have very interesting gut microbiome, but they live a very interesting life and they eat a very interesting diet. So part of the year they eat meat and hunt for animals like baboons. It's super wild. And this other part of the year they just eat fruit and honey. So I'm curious how you think about the trade-offs, to get back to the original question of like, you need money and there's a convenience in just accessing the closest people, the people that are most readily available, but those might not be proportional to the population. So I'm just curious how you think about the composition of the donor network.
Yeah, how about let me get back to that one. And I like to repeat stuff. So I'll tell you again the 3 pillars that we have from research, and then we'll go back to that one, which is an addition. But as a quick recap for the listeners, so the first pillar again is we have a donor network. The second pillar is we're building a comprehensive collection of all known human gut bacteria. And the third pillar is once we have the microbes, we can start to understand what they're doing. So we've been able to turn the microbiome into a screenable system. And this unlocks a lot because really for the first time, we can take the microbiome and input it into any assay for biology. So this allows us to actually complement other fields where we can ask questions of, does the microbiome, the metabolites and the proteins these microbes are producing, influence, say, targets relevant for pain? And so those are the 3 pillars that are core of the research component of the company. And what we've leveraged this system for is to then identify products and opportunities and therapeutic areas that we may want to tackle. We've used this research to basically establish internal research programs as well as programs in collaboration with corporate partners. So our leading program internally is in depression, where we identified a single strain of bacteria designed to hit 2 targets relevant for depression that has efficacy comparable to the drug ketamine in animal models. We seek to first develop it as a consumer-facing probiotic. We then also have corporate partnerships, 3 of them that are active right now. One is with Unilever, in which we're mapping how food ingredients impact certain gut bacteria with functions of interest. The goal is to make food better. We have a collaboration with Kenvue, which is Johnson & Johnson's new consumer arm. This is to create a novel infant probiotic, a bacteria that in very large cohorts of people, kids that have it don't really have allergies and don't develop atopic dermatitis. We should put that back in these kids. And then the third is we have a program with a major pharma partner in pain. And this is super cool, this program, because what I talked about with like mapping things, we took a big panel of human gut bacteria, screened it against 20 different targets relevant for pain, and basically created a matrix of how do bugs influence pain targets. So like this is the practical process to get there. And then it's just a matter of commercialization, which at first we're looking at teaming up with corporate partners. So at a high level, again, we go from people to bugs to screening to product, and we commercialize a B2B with corporate partners. But now back to your question of diversity. I agree, it's totally important. If we know humanity has 10,000 species of gut bugs, as an example, not all of those you can get in, Boston. So we're aware of this. This is in part why I acquired a company last year called Asiabiome. So Asiabiome was out of Hong Kong, and they had a donor network of around 8,000 people from Hong Kong. These are novel samples. It's very hard to get these samples. And so we acquired them specifically to get diversity as part of our cohort. We have targeted collection efforts to also include diversity in America in every sample that we're getting. We also have active collaborations. We don't have to build everything ourselves. So we have active collaborations from a donor sample perspective in the works with some very, very large cohorts. And the goal is exactly as you said, to capture this diversity broadly. But this also leads you into a very interesting regulatory question too. So like certain societies, there's appropriately so protections against working with, say, specimens from like the Hazda tribes and using that commercially, because there's this ethical conundrum of like, even if there's something really good for society there, you're still potentially mishandling that communication with these groups. And so one thing we're very cautious of as we're on the quest for diversity, we're doing this in a very appropriate way where we have full consent of the samples, everybody's informed of what's happening, and we're particularly careful as we think about some of these more delicate samples, if you will. But does that answer your question? We're on the same page. We're looking to get diversity. This is really just a matter of, again, execution, because the tech is there to find them.
I had an interesting thought, which is might be related to my question or unrelated, but it's interesting to think about symbiotic relationships. I remember as a kid looking at the encyclopedia, you usually see like the bird on top of the elephant, or like the little fish nibbling away at the big fish. But it almost sounds like this gut microbiome stuff is a symbiotic relationship. I'm curious to get your take on the mind-body connection and how you frame it.
Yeah, so a few thoughts. One, like, what are we? I argue we are just a big bag of protein-protein interactions in biochemistry. Microbes are just smaller bags of protein-protein interactions in biochemistry, right? And they're inside of us. So like, it's very obvious we're connected. When you look at many of the proteins we have, bacteria were the first ones to evolve them, and they still retain many of them. As you talk about like the gut-brain axis or mind-body connection, like this was early days of some of the stuff that I was working on. Think about like neuroscience. We study a lot around receptors that sense neurotransmitters as well as the neurotransmitters themselves. And we have proteins that produce those neurotransmitters. But if you make a list of different neurotransmitters and neuromodulators that we've been looking to drug for a long time, and then ask the question, can microbes produce or modulate those? The answer is largely yes. And, to me, it's like, we're really just on a spectrum of evolution. And we've landed on this place where we have us as the host, and then inside of us are where the microbes are happy. And Holobiome was actually named Holobiome around this concept of like a complete functional unit, where there's this phrase called holobiont, which is effectively used to describe a superorganism. And I'll take you down one path, which is depression. which is one of the areas we're very interested in. But, let's ask the question, is there, evidence to suggest the microbiome is involved with depression? So one way to look at this, specifically the gut, is you could say, all right, people with depression, do they have increased risk of like GI disorders? The answer is yes. Something like up to 75% of people with depression have GI issues. Okay. And next could be, what about known microbiome modulators? We know diet changes the microbiome, but we also know that we have drugs that take the microbiome, like antibiotics. So when you look at diet, bad diet, the Western diet, is associated with increased risk of depression, but the Mediterranean diet is the opposite. And one of my collaborators literally treated people with depression with the Mediterranean diet. It's called the SMILES trial. All right, diet, good evidence, that maybe there's a microbiome connection, obviously. Antibiotics. Now here we can get pretty, specific, right? So there's now cohorts, epidemiological cohorts of hundreds of thousands, millions of people, where the question is asked, if a person takes an antibiotic which kills the bacteria, versus an antiviral, which kills the virus, versus an antifungal, which kills the fungus, is there an increased risk of developing psychosis and depression? There is not an increased risk with antivirals. There's not an increased risk with antifungals, but there is an increased risk with antibiotics, things that kill bacteria. Okay, here we go. We're walking towards the GI issues. Yeah, okay. We've known that for a long time. Diet, yeah, we know that modulates the microbiome, also tied to depression. Antibiotics, okay, increased risk. Now you get to the mind-blowing stuff, where you can take the microbiome from somebody with depression, transfer it into an animal model of depression, and transfer depressive-like behavior. So, this is just a set of experiments that, are all hypothesis generating, in my opinion. But what we want to do is chase signals to find the real biology and then develop interventions. And if you go beyond what I just said of those four, and now you approach it from a mechanistic arena of like experts in depression and what they're studying, is there evidence microbes hit those same targets? The answer is also yes. So it's like screaming at me, where like maybe microbes are the reason in some people that they have depressive-like behavior.
Yeah.
And if that's the case, we get to the cause. We don't treat the symptom. And we can play that same game for many other diseases and associate them with the microbiome. So this is why I said before, there's all these signals, even if 10% of them are true. We could be curing depression in certain people. We could be preventing Parkinson's.
Yeah.
And in terms of drivers, like we have to do this as a society, and we have to have an open mind. Getting a little personal, again, drivers. A couple of weeks ago, I went to the land of cows and cornfield because I had to attend a funeral because someone who I love deeply killed themselves. On the way to the funeral, I stopped at a memory care clinic where my stepfather is currently residing because he has Lewy body dementia. And, me and my field, when my stepdad was diagnosed, I went everywhere to try and find a solution. And the top scientists in the field and the people funding big companies in the arena of dementia, they told me to take care of my mom. We got to be better, right? And we got to, challenge things. We got to move faster.
Yeah.
We can do this responsibly. But anyways, that answers your question from the mind-gut connection, I hope, and maybe a little bit broader too.
That's incredible. I'm really hitting stories and analogies and examples. Maybe curious to take that one step further. It seems like one of the indicators of how it influences or modulates is through these chemicals like dopamine, norepinephrine. How are we connecting the gut and the brain? And are there any like clear connections besides just sort of these experiments? I think you've also said once that 1 in 5 people will be diagnosed with anxiety and 1 in 10 will be with depression. And, it's a huge problem. Have you gone there with Holobiome?
The dogma at the moment, or what we think is happening, you have bacteria, say, in the gut. They could produce metabolites or proteins that make it into the bloodstream, some of which can make it into the brain and have a direct impact. There's good evidence now that microbes communicate to the brain directly via the vagus. And how they do that is, as you think about like your gut, it's like a tube. And on the perimeter of the gut, you have different cells. So some cells are, say, involved in like mechanical things to actually pass food through the GI tract. Some are involved in like sensing what's there. There's a set of cells called enterochromaffin cells, aka neuropod cells. These are found by, or really studied by one of my buddies, Diego Borquez out of Duke University. Basically, this neuron lives in your gut, senses stuff in the gut, is hardwired to the vagus, and the terminal neurons in the brain. And so, you have a few different paths of how this can happen. And then the key thing is figuring out, like, how does that crosstalk occur? What's the chemical messengers? What's the pathways? There's evidence that certain immune cells can sense in the gut and actually translocate to the brain and influence things like neuroinflammation. So again, this gets back to, we're just like big bags of protein-protein interactions in biochemistry. And like, there's some channels that connect different bags that have barriers. But like, we're starting to just understand this so much better. So this is a very process-oriented approach. And we have undertaken that process-oriented approach for multiple programs. So our depression program is ready to go into manufacturing and human testing right now. Our pain program is still in the discovery phase, but we're very optimistic that's going to come through quickly. Our partnership programs are looking really good. So, we think we figured out that part. And we've also built the research component, which the real dream is that we can continue to spin the wheel on the research component to map this stuff. And just like in the Genzyme days, I'd love to actually work directly with patient groups. So have them as part of the entry point and just run them through an efficient microbiome machine to solve it. And then on the other end of it, we have a very streamlined commercialization path to just get it out there and then learn to close the circuit. From a funding perspective, we brought in mainly non-dilutive grants and corporate partnerships. So Holobiome's brought in actually quite a small amount of venture capital. 80% has been non-dilutive. And we have, leveraged these corporate partnerships efficiently.
Yeah.
But yeah, it's been a fun ride.
That's awesome. Phil, it's clear you approach your research, your company with a lot of integrity. You are scaling. You have a platform to produce really great consumer and also pharmaceutical solutions. I just want to give the floor to you because I really appreciate learning from you this whole time. And I think I would love for everyone else to hear what you've got in store and what we can do to help.
Yeah, of course. I mean, like I said earlier, I'm not here by myself. I have an incredible team. And that they've really stuck with us, which is awesome. And so like for anybody listening that's wanting to get into biotech, like find your people that you want to hang out with and grow with. And again, don't be an asshole. From like an ask perspective, I mean, if you like this science and you're interested in it, like reach out and, would be happy to chat with anyone. I get emails all the time from like patient groups, individuals. I try my best to respond to them. But, we got to do this as a community. Biology is getting there. And the technology is here to do it right in microbiome. And this fire I have, my team does too. And we're surrounded by a bunch of folks in the same camp. We're working as hard as we can. So I think it'll get there. And I'm excited for that'll look like. So there we go.
I am as well.
Amazing. Me too. I can't wait. I'm waiting to eat these off the shelves to get these plants and to fill up my garden again. This is amazing.
There you go. Yeah, no, this was a lot of fun. Really good questions too, by the way. Like that was great.
Appreciate you. If you enjoyed that episode, please like and subscribe. We're just getting started to tell the stories of these amazing creators, athletes, and entrepreneurs. Drop a comment below on what was your favorite part of this episode, and stay tuned because we're going to be releasing new episodes every 2 weeks for On the Rise. See you next time.